Go. Okay, good morning, everyone. So, I'm not as tall as Sharaf or Ashraf. I'm not as tall as you, so they have to lower it down. So, good morning, everyone. So, my slides will be in English. I will be talking in English, but please feel free to interrupt me anytime. I can answer your questions in Arabic or in French, as you wish. So, before I start, big thanks to the University of Tlemcen, Dr. Gawar in particular for this, great opportunity. It's a great opportunity for me as well to share my work with you guys, but also to learn from you and with you.
Thank you.
I think you are doing a great job.
Thank you.
Okay, so just before I start also, thanks Ashraf or Sharaf, because you went through all the introduction I didn't want to go through. So I will skip all the introduction about the technology and the sequencing and everything. I'm not working in the biodiversity field, but as you all know, it's a very important field that is related to everything, human health, animal biodiversity, and plant biodiversity as well. Okay, this is just quick disclosures. I will skip this. Quick summary about my background. I am from Algeria, of course. I did my undergrad in computer science. I went to Montreal, then from Montreal to the United States for my postdocs in Florida and Cleveland, Ohio. In 2015, I landed in, it's going very fast. Give me 1 second. Okay, 2015, I landed in Boston at the Harvard University where I have my lab now. Quickly, because I have this hybrid background as a computer scientist, also through my PhD, master's degree, and my postdocs, I was introduced to the field of immunology, virology, and molecular biology. I am very blessed because I can handle projects that use both techniques or both research fields to answer very highly relevant questions related to human health in general, disease pathogenesis, vaccine development, that can be applied to some extent to plant and animal biodiversity or studies. So this is just a snapshot of what I do in the lab. Mainly, I am using omics techniques to investigate how the host and the virus interacts, what we call the host-virus interface, using a multi-omics approach, including metabolomics, glycomics, transcriptomics, and proteomics. We are also interested in looking at the epigenetics of the host and how viral infections or viruses affect epigenetics, but also how the epigenetics is important to fight some viral infections. Epigenetics are the changes in the chromatin in contrast to the genomics or the transcriptomics that is more about the genes. Another aspect of my research is vaccine development. So we are interested in developing vaccines for emerging viruses, including one of the most deadliest viruses we have seen, COVID-19, for instance, but other viruses such as TB, Zika, monkeypox, and we are doing some work on cancer and HIV. And lately, and because, as I said at the beginning of my hybrid background, I'm trying to incorporate AI and machine learning in some of the omics work we do. And if you are familiar at least a little bit with omics, you know that these are huge data sets. We cannot just use standard coding or standard pipelines to analyze it. We need at some point to incorporate AI and machine learning in that. Today's talk is about harnessing the power of omics technologies to study the host pathogen interaction and the host immune response to vaccines. And I will skip most of the intro slide, as I said, but for this one, I just want to highlight that omics is not only about looking into human health or vaccine development or understand how virus affect a human being or its his or her immune system, but it's also about looking into biodiversity. It's about also some security-based application and even now there is this new field of biofuel using algs, or what we call the algomics, where we are trying to generate energy from algae because we want to protect the environment, of course. Okay, this is also another snapshot of the big picture of an omics project. This is not exclusive. This is not the only things we need or we do, but mainly when you want to start an omics project, it starts by hypothesis, an idea. What do you want to do? What are you trying to answer or to investigate? And as you can see on this schematic, you can do omics, any omics project using human data, animal models such as monkeys or mice or any other animal models, of course, but also from plants. So first of all, you need to specify your hypothesis. What do you want to do and how do you want to do it? You collect your samples, whether it's a one-time point or a several time points. Then you need to define what kind of assays or lab work you want to perform, you want to do, right? And for that, as Dr. Sharaf explained it very well, there are different techniques, there are different assays for omics work, whether you want to look at gene expression or the transcriptome. You can do bulk or single cell RNA sequencing. If you want to sequence DNA, it's usually NGS. And if you want to do proteomics, looking into the full proteome of the host, there are different techniques. And for now, one of them, among the most known techniques for proteomics profiling is mass spectrometry, of course, SomaScan technologies that can investigate 11,000 proteins, and there is another technology called Olink that can also explore thousands of proteins. You can also look into the metabolomics because it's important to understand the metabolites in the context of viral infections or immune vaccination, and glycomics, looking at the sugar on the surface of the cells, which are also very important for the way the immune system or the host respond to viral infections or even to vaccines. The last part is about, now that you defined your hypothesis, you know what to do, you know how to do it, what kind of assays you need, you generated your data using this omics or this bulk or single cell or NGS techniques, then what? You need to analyze the data, right? And these are billions and billions of millions of data sets. You cannot just analyze it using your own computer at home. You need an infrastructure and you need sophisticated tools and pipelines. And this is where bioinformatics comes in handy. Why? Because you need a bioinformatician or a data scientist or a computational biologist who not only knows how to code, how to write the code, to analyze the data, but also someone who understands the hypothesis, the biology. If you are a bioinformatician and if you are a computer scientist, you code very well with R, with Python, or any other language, you can do bioinformatics, but please go and take some courses in biology, immunology, or virology, because you need to understand how the cell, at least the basics of how a cell works, right? How the immune system works. And vice versa, if you are a biologist or even a medical doctor and you want to do some bioinformatics, go and learn some coding techniques such as R or Python, at least. And now with the, now that AI is kind of becoming the hot topic of the week, and we all know what AI can do. If you have used ChatGPT in the last few months, you know how much AI is very important and how much AI can help. So now we are trying to incorporate some AI-based techniques in our omics data analysis. Okay, so the second part of my slide, sorry, before I move to the next one, it's just a summary of, again, it's not an inclusive list, but it's just a summary of some of the techniques and some of the softwares. Feel free to take a picture, feel free to ask me if you want. These are some of the tools we use in the lab to analyze our omics data set, whether it's transcriptomics, proteomics, glycomics, or metabolomics. These are some of the tools we use to generate the data on the first row, single cell, bulk-seq, proteomics, etc. And here you have the name of the tools or the pipelines. These are, for most of them, if not all of them, free of access. Free. Whether if you are familiar with if you have some basics in coding or in computing, you can go to GitHub. Most of them have a GitHub web page. You can just download the tools, or you can just Google the name of the tool, and they are free for you to use. And the beauty for someone who wants to learn bioinformatics now, and this is just something entre parenthèses, you can learn bioinformatics without generating your own data. Why? Because there are also free databases you can use for your own learning or teaching. These are, for instance, all the papers, the omics-based papers we have published as a lab are now put available in a database called GeoOmnibus. You can access this data for free. If you cannot reach out to the authors of the paper, just send a friendly email saying, hello, I'm interested in using your data set as part of my training in bioinformatics. Can you give me access to the data? And we are required to share this data with you, because when we publish a scientific paper, it's an obligation. We are required to make the data public for everyone, everywhere in the world. So if you are interested in learning bioinformatics, it's easy for you as students. I'm talking to students mostly. You can have the tools, most of them are free, and you can also download the data. Okay, so one of the big challenges, and bioinformatician in the room, I hope they will agree with me, is data integration. You know, biology is not about one cell, it's not about one protein or one gene. Biology is about the whole system and how this well-synchronized immune system is working, right? So we cannot just analyze omics data as separate groups or blocks or hubs. At some point, the role or the job of a bioinformatician is also to find the best way how to integrate this data together in some kind of a network. It's a network. Why? Because we know that genes do not work alone. Proteins also do not work alone. They all collaborate. They are all connected to each other, and they co-regulate each other. So if you are doing an omics project and you have generated, even if you are in the biodiversity field actually, and you have generated this proteomics and gene expression and epigenetics, you need to at some point to think of the best way of integrating this data. Why? Because a gene is affecting the protein, a protein is affecting the epigenome, etc. And it's also affecting the metabolites, right? So data integration is one of the big challenges in the omics field in general. There are tools, there are ways of doing data integration, but it takes a good understanding of the biology and how these different components are linked or correlated with each other. Now, I'll start by, I don't want to focus on one project, so I'm going to give you like snapshots, one slide, two slides about some of the projects we have published, we have conducted in the lab about the use of omics technologies for specific questions, right? So the first one is about Zika, and this is one of the, actually this was one of the first omics project I led in the lab. The paper is published in the Cell journal in 2016, and it was about Zika. Why Zika? I'm not sure if you are familiar. I hope some of you are. Zika is a virus that is, the reason of this viral infection is a mosquito, une mouche, in specific places or countries in the world, not everywhere. I'm not sure if Algeria had this, the Zika pandemic back in 2016-17, but it was a big deal. Why? Because when a human being, a normal human being, is infected with Zika, usually after 2 weeks, the virus is cleared and no harm is left. However, in pregnant women, Zika is very dangerous. Why? Because when the virus passes from the mother to the fetus gets this, what we call these issues in the brain development. I will not show you the pictures of how the Zika virus is damaging for babies, born babies with very small head, with a very small brain, most of them die afterwards, but even if they survive with big issues related to the brain development. And you all know that if your brain is not developed, there is nothing you can do much, right? So this is the reason why we kind of were interested in looking into the Zika back then. So what we did, we cannot do Zika investigation on human beings, of course, so we used monkeys, rhesus macaques. So we infected rhesus macaques with Zika. We collected samples from blood and different tissues, and we looked at when you get Zika infection, what happens, right? What are the pathways? What are the genes? What are the proteins that are modulated by the virus? And how long this dysregulation lasts? And are there any, not side effects, but are there any things left after the virus is cleared from the blood? So as I said, the paper is published, so I'm not going to go through the details, but we were able to find that after Zika infection in a rhesus macaque model, there are some specific pathways related to inflammation. So viral infection is about pro-inflammation, of course, and one of the specifics of Zika infection is this increase of pro-inflammatory pathways, and also specific pathways, which is the mTOR pathway or the mechanistic target for rapamycin. The second conclusion is we all know that Zika is creating or generating these issues in the brain. So we were able to find that in Zika infection, there are issues with what we call the extracellular matrix, the matrix extracellulaire, which is the matrix, it's important for the movement, for the way cells go around the body, right, or within the cell. So we were able to find that Zika virus or Zika infection is creating openings or breaches into the blood-brain barrier. There is a barrier between the circulation and the brain. Why? Because brain is what we call well-controlled environment. Almost nothing is authorized to go into the brain in a more like open way. Everything is well controlled because of this barrier, right? But Zika virus actually creates these openings or these breaches in this blood-brain barrier, leading the virus to the brain. This is very important. So we know that the virus gets into the brain by creating these issues into the barrier. Imaginez une barrière sécuritaire et le virus est comme un agent terroriste. Il crée des petites ouvertures pour s'infiltrer et aller infecter le cerveau. A second project, so these are the major conclusions of the paper. Now, the second project related to vaccination. So you all know about COVID-19 and how deadly the virus was, at least in most countries, actually, even in Algeria. So we wanted to investigate how COVID-19 or SARS-CoV-2 affects the immune system, and what are the pathways again, or the proteins, the genes, or the epigenetic changes induced by this infection. We don't do that in humans. We do it in animal models to understand, because our goal is to understand how, not why, but how it's happening. So in this case, we used both the macaque model and hamsters. Hamsters was a good model to study SARS-CoV-2 because we showed that when you infect a hamster, you get severe disease similar to what we get in human. So keep in mind, when you select an animal model to study viral infection, you need to make sure that this animal model mimic what's happening in human. So we selected hamsters in this specific case because hamsters get very sick when they are infected with COVID-19. So in this paper, again, we infected, we took two groups of hamsters. The first group was vaccinated. We gave them the I-26 vaccine, which is the Janssen vaccine. And the second group, they were not vaccinated. They are sham or control groups. Then we gave them the virus. We infected them. And we found that when you get the vaccine, animals are protected about developing severe disease. So they get the virus, they get the infection, but they don't get very sick, they don't lose weight, and they don't die. So the conclusion of this is that even though COVID vaccines are not perfect, they are not protecting 100% from severe disease or from infection, but it's better to get vaccinated rather not, because you will be protected to some extent about these severe symptoms of COVID-19. The other project related to COVID-19 is what we call long COVID. Long COVID, most of us never heard of. Why? Because long COVID is something 10 to 20% COVID-infected individual develop after a COVID infection. You don't realize it, you don't know it, because the symptoms are shared with other viral infections. You are sick after a COVID infection, you have brain fog, memory loss, you can't exercise, you can't work, you can't concentrate, you can't focus. Why? And you might think that, well, I'm just sick, my immune system is down. But I'm not saying that 100% is due to COVID, but now it's a fact. 10 to 20% of individuals who get COVID-19 develop what we call long COVID. It's a real issue, it's a real health issue, it's not a joke, and many labs, including ours now, are working on trying to figure out why these 10 to 20% of individuals develop long COVID, and if we understand the why or the how, we can think of maybe some therapeutics. So the schematics on the left, on the right, are the range of symptoms some of these people with long COVID have. And if you look at the list, actually, most of these symptoms, you can have them after any viral infection, right? So that's why it's hard to distinguish long COVID from something else. The symptoms are mainly common to most viral infections, but 10 to 20% of individuals with COVID infection have it. And one of the major reasons of this high interest at the high level for this long COVID issue, and there is actually at NIH in the United States, which is the National Institute of Health, is pouring a lot of money in this research, because you want to find cure, we want to find a drug, we want to find a medication to these people who are suffering from these long-lasting symptoms of COVID. Why? Look at the range of tissues that are affected by long COVID. Long COVID is not only a matter of affecting the lung or the respiratory system, it can affect the heart, leading to cardiovascular disease, the kidney, a lot of issues with kidney, memory loss, and again, these are all speculations. We don't have real data to support it, but there are some evidence that long COVID is somehow involved in Alzheimer's disease. And it's kind of understandable, because if long COVID is affecting the brain and the memory in some way, it could be also related to Alzheimer's, right? So this is the reason why we are highly interested in looking into that and trying to find a cure, hopefully. Another aspect of the work we do using these multi-omics techniques is sex bias in response to vaccination. We tend to think that a vaccine or a medication, whether you give it to a man or a woman, is the same. It's not, right? Why? Because our immune system is not the same. We don't respond the same way to viral infections. We don't function the same way. So for COVID vaccination, actually, we have an ongoing study where we showed that the pathways that are activated or not in men versus women vaccinated with different COVID vaccines are not the same. So, for instance, in women, we found that there is an increase in interferon type 1, right? Which at this point doesn't mean much, because this is still very early. But just to tell you that this is something we need to keep in mind as omics researcher, or even, you know, for an immunologist or a bioinformatician or virologist or even a doctor, women and men, I mean, the immune system of female and male are not the same, so we don't respond the same way to viral infections, and we don't respond the same way to vaccines. We are not even protected the same way. Okay, so this is also another project where, you know, we tend to look at always after vaccination, after infection, right? You get infected, what happened after infection? What happens after vaccination? How about the baseline? How about your natural immune system? Before getting the vaccine, before even getting the virus, we know that, again, we know that we have, some of us have a predisposition to get infected quickly, others have a predisposition to fight the infection or to resist the infection better. Why? Because of the way we are, of our baseline immune system. So this is another research aspect of the work we do in the lab. We are trying to investigate what are the baseline correlates, we call them correlates, of a good vaccine response or a better response to a viral infection or control of infection. Why? Because if we are able to put like a name on these proteins or these genes that are important to make you protect better if you get infected, it's important. You can use these genes or proteins, make it a medication or a vaccine, and give it to these people who cannot do it, who doesn't have these genes, who do not have these genes or proteins in their, you know, as part of their baseline immune system. So this is also something we are highly interested in. Back to the omics technologies, and quickly, I want to touch base on what we call bulk versus single cell. If you look at gene expression, or what we call the transcriptomics profiling, in humans, we investigate the mRNA or the expression level of 30,000 transcripts, let's say around 20,000 genes. You can do it using two techniques, at least two techniques. One of them is bulk RNA sequencing, and the second one is single-cell RNA sequencing. And I like this picture to depict that depicts the differences between the two. Bulk RNA sequencing, you are looking at the expression of your 20,000 genes, to make it simple, as a bulk. You do not know which cell is expressing this specific gene. You are looking at the absolute value. It's like the median of the expression of these genes in all cells that express the gene. But the single-cell technology actually is an amazing technology, by the way. It gives you the expression of these 20,000 genes in every single cell. Imagine you take a blood sample or a tissue sample, millions and millions of cells, and you can investigate the expression of 20,000 genes within every single cell using this single-cell technology. It's just an amazing one. It's a very expensive one, however, and this is also a snapshot of the cost. On the left is the single cell, on the right the bulk-seq, and you can see the differences in the price. Not only you need to have the instrument, which is not expensive, by the way, but because you get it once, but the reagent, the sequencing, the analysis is very expensive, and basically doing one single cell, one sample by single cell is around $3,000 to $5,000. So it's a very expensive technology still. Now, we are using these two technologies in the lab, and this is the reason I wanted to introduce you to this single cell. One of the projects actually we investigated using single cell is looking into the cell. You know, when you have an immune response, it's not just about one cell population, it's about a group of cell types or cell populations. And this is the reason why we use the single cell to look at the immune cells that are activated by a vaccine at different time points. On my slide, I am showing some data from a project where we collected samples, cells or blood from vaccinated individuals that received the I-26 or the Janssen vaccine. And we did single cell to investigate the different immune cells that are activated by the vaccine. On the left, you see what we call the UMAP or the PCA. This is the plot you get when you do a single cell. Every single dot on the figure is a unique cell. Okay, just imagine the extent of this data set. Every single dot is one cell, and for every single cell you have the expression of your 20,000 genes or 30,000 transcripts, whatever you are looking at. Now, on the left, on the second part of the plot, we annotated, we put names on these dots. Each color represents a cell type, as shown on the figure, showing activation of T cells, activation of B cells, activation of some innate immune cells, such as macrophages, monocytes, etc. So this is the power of doing a single cell project. Another application is doing what we call longitudinal profiling of this immune activation. On the left, you have the same analysis for day 64 days after vaccination. And on the left, you have the same data, but 239 days after vaccination to show the evolution, how this immune activation of immune cell activation happens after vaccination. And you can track your immune population from day 1 to day 2 to day 3, etc., depending on your experiments. And this is all something you can do using this single cell, this amazing single cell technology. I think this ends my talk. This is a picture of our lab took last summer at our lab picnic. Every year we have a picnic. All the lab gets together for one day. And yeah, thank you so much.
Thank you, Malika.
My pleasure. Thank you.